Hi guys, welcome back to another video from BioBuddy. So in this video guys, we will be talking about cloning. As you all must have studied what are clones. Basically, clone is a simple word which means an identical copy, right? We know that in CDNA cloning, what are we cloning? A CDNA. CDNA stands for the complementary DNA. Right. In the previous videos guys, I've shown you how the cDNA synthesis is taking place, that how is this cDNA being synthesized first from a primer mRNA strand, and that mRNA strand is used as a primer, right? From that the cDNA strand is generated, the single cDNA strand is further duplicated, and then it is cut with the help of S1 nuclease enzyme. I think this previous video has been uploaded. Please guys watch that video and then come back to this one, because then this will be more simpler. So in cDNA cloning is one of the major tools in molecular biology, because it is helping us in doing what? In preparing clones, right? It is helping us in the preparation of clones of the same DNA which we have been prepared through the synthesis method, and now we are cloning that method with the help of, and preparing their identical copies with the help of cDNA cloning. So in this method, what we are doing, we have taken a vector, but this vector has to be cut from a specific site, and this vector is cut down by restriction endonuclease, but we are using restriction endonuclease type 2, right? This is the most common restriction endonuclease which is being used here. So now what is happening, this vector which is present here, it is cut down by the restriction endonuclease type 2 from a specific site. This is a specific site, right? Now this is the chromosomal DNA which we require. Like we require something, for example, antibiotic resistance, right? So it can be tetracycline. So if we want it to be tetracycline resistant, so we will take out that portion from that particular DNA. So now this chromosomal DNA fragment has to be cloned. So we cut down this chromosome from the entire DNA fragment. We are cutting out only this fragment, and this is also done by restriction endonuclease. So restriction endonuclease here is playing an important role in order to cut the proper DNA fragments or vectors from their specific sites which are to be cloned, right? So this one is being cloned from this site. Now both of them have been cut from their respective places, and now they have to be together joined. For joining, you also know, for joining we use DNA ligase, but in this case we are using a specific DNA ligase. What is this DNA ligase? It is T4 DNA ligase. T4 DNA ligase is being used here specifically because it is being extracted from E. coli bacteriophage T4. This E. coli bacteriophage T4 is being used to extract out what? T4 DNA ligase. Now this T4 DNA ligase will further be used to join the two segments, that is one segment which we have been extracted, the chromosomal DNA fragment, and the other, that is the vector to which we have to join it. दोनों को join करवाएगा. Now what is happening here? In this you can easily see, this is our vector cut down by restriction endonuclease type 2, and this is our chromosomal fragment which needs to be ligated to that vector, so that more and more copies of that particular segment can be generated. So this is done by DNA ligase, and specific DNA ligase is mentioned here. The name T4 DNA ligase is being used in this case. So you have seen this dotted structure means that this portion is being cut, right? So now the cut portion is attached to this by DNA ligase, and now what we are using, we are taking Mg2 plus ions and ATP. ATP is a form of energy, that is adenosine triphosphate. Here adenosine triphosphate is providing much energy for the DNA ligase to help in the joining of both the segments. Now when these segments with the fragment and this chromosome are vector are joined together, this will again be formed a complete circular form like that of the vector. Now this vector will again be put inside the host cell. This is known as the host cell, which is having its own plasmid. This is the host cell, and now what we are doing, we are adding this clone part into the host cell mechanism so that they take use of their whole machinery and start implanting their own processes. So that way now the daughter cells which are being prepared by this organism or the host cell, the daughter cells will have the identical copies of this rDNA which is being added. Why? Because this rDNA will take up their machinery and make up its own and help in the, अब क्या होगा, जितना भी हमने इसके अंदर मटेरियल डाला हुआ है वो पूरा का पूरा यही रेप्लिकेट कर जाएगा, और रेप्लिकेट करने के बाद डिवाइड करके जो भी कॉलोनी फॉर्म होगी है, जो भी डॉटर सेल्स फॉर्म होंगे, उसमें कौन सा डीएनए होगा, यही डीएनए जो हमने क्लोन किया है. तो दिस वे this cloning is being done, that is the production of individual copies which are similar in nature, जिनका size और similar है. So this is how DNA cloning is very important for molecular biology, right? इसमें क्या होता है, hybridization से easily it can be done by ligation, and which ligation specific this one is being used, T4 DNA ligase, which is extracted from E. coli bacteriophage T4. All the details are mentioned here. Mg2 plus and ATP are playing an important role here, because DNA ligase also requires some kind of energy to do the function, right? So this energy is being taken up by them, and right now in this process only this much is required, guys. This is a very simple video regarding all these things of CDNA cloning, and if you haven't seen the CDNA library, I will be making a video on the CDNA library as well. So CDNA synthesis is already prepared and genomic library is also prepared, so go guys, please watch those videos and then you can come back, because this is very simple. It is a normal complementary DNA synthesis cloning जो नॉर्मली भी होती है वैसे भी होती है, बट इसमें हम अलग क्या कर रहे हैं, इसमें हम CDNA स्ट्रैंड को ले रहे हैं अलग से. दैट्स इट, राइट? सो आई होप यू गाइज लाइक द वीडियो. Subscribe to my channel guys, like my videos, comment down below for any doubts. Thank you, keep watching, stay tuned for the next video.